You know, when we think about the brain, neurons get all the attention, don't they? The signals, the connections.
You really do.
But today, we're doing a deep dive into something just as vital, maybe a bit more behind the scenes. Oligodendrocytes. We've got some great questions guiding us.
Yeah, they're absolutely crucial players.
Our mission, then, is to really get what these cells are and why they're so fundamental for, well, everything our brain does.
Exactly. You can think of oligodendrocytes, or oligos for short sometimes, as the specialized electricians of the central nervous system, brain and spinal cord.
Electricians. Okay, so they're glial cells, right? Not neurons.
Correct. Glial cells. Their main role is support, but crucially, insulation for our nerve fibers, the axons.
Insulation. Okay, that leads directly to myelination, doesn't it? What exactly is that?
Myelination is the process. It's how these oligodendrocytes wrap around the axons with this fatty substance called myelin. It forms a sheath.
Like wrapping electrical tape around a wire.
That's a pretty good analogy, yeah. It prevents the signal from leaking out and helps it travel efficiently.
Now, I know there are also Schwann cells. They do something similar.
They do, but it's a key difference in location and technique, you could say.
Right.
Oligodendrocytes are only in the central nervous system, brain, and spinal cord. Schwann cells handle the peripheral nervous system, all the other nerves.
Ah, okay. Location and technique.
Right. A single oligodendrocyte is quite efficient. It can wrap its myelin around segments of, say, 30 or 40 different axons.
Wow, okay. Multitaskers.
Very much so. Whereas a Schwann cell dedicates itself to just one segment of one axon in the periphery. Different strategies.
Fascinating. So these CNS electricians are really covering a lot of ground. How much does this myelin actually speed things up? Is it a minor boost or?
Oh, it's dramatic. Absolutely dramatic. We're talking signals jumping from maybe a few meters per second on an unmyelinated axon to potentially 100 meters per second or even more on a myelinated one.
Whoa, that's huge. Like you said, dial-up versus fiber optic.
Precisely. The signal effectively hops between gaps in the myelin, the nodes of Ranvier.
Yeah.
It's called saltatory conduction, super fast.
Right, right. So it follows that if something damages these oligodendrocytes or their myelin, the consequences must be severe, like in multiple sclerosis.
Exactly. MS is, unfortunately, the classic example. The immune system mistakenly attacks the myelin sheath and sometimes the oligodendrocytes themselves.
And that disrupts the signals.
It disrupts them massively, slows them down, blocks them sometimes. This leads to all the varied neurological symptoms you see in MS: weakness, vision problems, coordination issues, fatigue. It really depends on where the damage occurs.
Devastating.
Yeah.
Where do these cells originate? Are we born with all we'll ever have?
Not quite. They develop from specific precursor cells called oligodendrocyte precursor cells, or OPCs. During development, these OPCs multiply, travel to where they're needed in the brain and spinal cord, and then mature into the myelin-producing cells.
So there's a production line, essentially.
You could think of it like that, yeah. Understanding that process is really important for thinking about repair.
Which is the big question, isn't it? If they get damaged, say in MS or after an injury, can the brain repair them, make new ones?
That's the million-dollar question and a huge focus of research. There is some capacity for natural repair remyelination. Those OPCs we mentioned can sometimes activate, mature, and lay down new myelin.
But it's often not enough.
Often not enough, unfortunately, especially in chronic diseases like MS. The repair process can falter or become inefficient over time. So researchers are desperately trying to figure out how to boost that natural repair.
Are there signals involved, like instructions telling the cells when to make myelin or when to repair?
Oh, absolutely. It's a very complex environment. There's a whole soup of signaling molecules, growth factors, inhibitors that regulate whether OPCs mature, whether oligodendrocytes survive, how much myelin they make. It's tightly controlled.
And what about other kinds of brain injuries, say it's a stroke or trauma? Do oligodendrocytes react then?
They do. They're often vulnerable to the secondary damage that happens after an injury, things like inflammation, lack of oxygen. So they can be lost. But they and the OPCs are also part of the brain's attempt to respond and potentially repair afterwards, though, again, often imperfectly.
It really sounds like finding ways to protect or replace these cells could be revolutionary. Are new therapies targeting this?
Yes. Definitely. This is probably one of the most exciting areas in neurology right now. There's a lot of work on therapies aiming to promote remyelination.
Like what kind of approaches?
Well, strategies to encourage those OPCs to mature into myelinating cells, drugs to protect the existing oligodendrocytes from damage, and even more experimental ideas like cell transplantation. It's moving fast.
That's genuinely hopeful. So let's recap quickly. Oligodendrocytes, the crucial insulators of our central nervous system,
Wrapping axons in myelin.
which makes nerve signals incredibly fast, essential for basically everything.
Right. And damage to them, like an MS, causes major neurological problems because those signals get disrupted.
But, and this is the hopeful part, there's a lot of research focused on repair, on stimulating those precursor cells, on finding ways to rebuild that vital myelin.
Exactly. The potential for future treatments is significant.
It really makes you think, doesn't it? These seemingly small cells have such a gigantic impact on our health, our abilities. What other tiny components inside us are playing these massive roles that we're maybe just starting to appreciate?
That's a great point. It underscores the incredible complexity within us and how much more there is still to discover about how our own bodies work. There's a whole universe inside.